Hello students, so today the topic I'm going to discuss that is glycolysis. Okay, so you know that glycolysis is a common pathway of both aerobic and anaerobic respiration. Whether the process is taking place in the presence of oxygen or in the absence of oxygen, this glycolysis process take place in both of the cases, right? So what is the meaning of glycolysis? Basically the glycose term means sugar and lysis means water splitting. Okay, the glycolysis process is basically, it is also called as AMP pathway as well. What is the reason of it? That here E stand for the Gustav Emden, M stand for Otto Meyerhof and P stands for the Parnas. These three scientists were there who gave the scheme of the glycolysis, right? So here the glucose is the respiratory substrate which will be taken in the glycolysis, and the partial oxidation of the glucose will take place over here. So if I talk about the plants, in the plant's case the glucose is basically derived from the sucrose, and you all know that in the plant body the food is translocated in the form of sucrose. So what happens over there? The sucrose, first of all, it is broken down into the glucose and fructose in the presence of this enzyme that is called as invertase, and these both enters into the glycolysis pathway. Let's start the glycolysis pathway now. So first of all, we are taking as the glucose is the simplest form of sugar which will take part in the glycolysis, and you know that the glucose is basically, it will enter into the cell through the glucose transporters, right? So once the glucose enter into the cell, it will further go into the glycolysis process. You all know that glucose is a stable molecule. First of all, we'll have to make this molecule reactive, unstable, right? So first of all, in the first step we will do what? In this first step we will convert the glucose into glucose 6-phosphate, and the enzyme which will work over here, that is hexokinase, and Mg+2 magnesium ion will be working as a cofactor over here, right? In the first step we'll have to spend the ATP molecule because we are doing the phosphorylation of the glucose, so we'll have to break one ATP molecule over here. So in the first step you have to spend one molecule of the ATP. The ATP will break into what? It will break into the ADP, and the inorganic phosphate will go and it will attach on the glucose, and as a result the molecule that will be formed over here, that is glucose 6-phosphate, right? Here in case of the plant, sucrose can also break into the glucose and the glucose can enter into the glycolysis pathway. Sucrose also break into fructose and fructose also enter into the glycolysis pathway in between, right? So glucose 6-phosphate is formed over here. Now the glucose 6-phosphate will get isomerized into the fructose 6-phosphate. Now one question come over here, that's why we are isomerizing the glucose 6-phosphate into the fructose 6-phosphate. What is the reason of that? The reason is that the glucose molecule, if we see that the glucose molecule have only one side chain available where we can attach the phosphate group, but the fructose is having two side chains available where you can attach the phosphate group. One is here and one is here, and the first position and the sixth position. That's why we can do the phosphorylation again also. So that's why we are isomerizing the glucose 6-phosphate into fructose 6-phosphate, and the enzyme which is working over here, that is isomerase, right? The fructose can directly enter over here like this, and the ATP molecule will break, it will form the ADP, and the inorganic phosphate will go and attach on the fructose, and the resultant compound that will be formed over here, that is fructose 6-phosphate, right? Now we can again do the phosphorylation of the fructose 6-phosphate because it is having the two side chains available where you can add the phosphate group. Now you have added the phosphate group on both these positions, right? So ATP will break and it will form the ADP, and the inorganic phosphate go and attach on this compound. As a result, fructose 1,6-biphosphate is formed. The enzyme working in this process, that is phosphofructokinase enzyme, this is also called as the pacemaker enzyme. Now the question comes that why we are calling it the pacemaker enzyme. So dear students, you know that in our heart the pacemaker is there, that is SA node, called as sinoatrial node, and it is responsible for the generation of the action potential in our heart. In the same manner that the phosphofructokinase enzyme is the pacemaker enzyme of the glycolysis, because it is when the ADP and ATP ratio in the cell is different. For example, if the cell is having the more amount of ATP, it means the cell will not proceed this process further, and the ATP molecule will go and bind to the allostatic site of this enzyme, and the whole process will stop. But if the cell is having the high amount of ADP, it means the ATP amount is less in the cell, then the ADP will go and bind to the allostatic site of this enzyme, and the process will continue further. That's why this enzyme is called as the pacemaker enzyme of the glycolysis, and it's a kind of irreversible process. You can see in the diagram as well. So here also we can do the one phosphorylation, ATP to ADP, and inorganic phosphate go and attach on the fructose 6-phosphate, and the resultant compound that is formed, that is fructose 1,6-biphosphate. Now we have to break this fructose 1,6-biphosphate molecule into the two molecules. One is 3-PGL and one is DHAP. The meaning of DHAP is dihydroxyacetone phosphate, and 3-PGL means 3-phosphoglyceraldehyde, and the enzyme which is working over here, that is aldolase enzyme. Now I'm going to take that this enzyme, this molecule has continuing further, okay, and I'm concluding that this molecule is getting converted into 3-PGL. So further the two molecules will be taken in this pathway, right? So 3-PGL will get converted into 1,3-bipga, okay. In this process what will happen, two redox equivalents will get removed from this molecule, and it will be taken by the NAD+, and NAD+ will get converted into NADH plus H+. Two molecules of NADH plus H+ will be formed in this step. Here this NADH plus H+ is basically what? It is a reduced coenzyme, and when it go into the electron transport system or electron transport chain, it will be responsible for the production of energy over there, and that's why we are calling it oxidative phosphorylation. Okay, so NADH plus H plus will not give the direct energy, it will give the indirect form of energy which will be obtained in the electron transport system. In this step, the dehydrogenase enzyme is working over here, and the two inorganic phosphates are added over here. Okay, so the next compound that is formed, that is 1,3-biphosphoglyceric acid. Okay, so here you understood one thing, that NADH plus H plus is reduced to NAD plus is reduced to NADH plus H plus. So here reduction happened, but this molecule got oxidized. This molecule got oxidized. It means the aldehyde got oxidized and formation of acid taken place, right? 1,3-biphosphoglyseric acid. After that, 1,3-biphosphogylyceric acid will get converted into 3-phosphoglyceric acid. The enzyme which is working over here, that is phosphoglycerate kinase. Okay, here the 2 inorganic phosphate molecules will come out. It will be taken by the ADP. So you understand one thing, that 2 ADP molecules will get converted into 2 ATPs. This is called as SLP. What is the meaning of SLP? SLP means substrate level phosphorylation. It means here the substrate is releasing the energy, right? That's why we are calling it SLPS, substrate level phosphorylation, right? So the next compound that is formed is the 3-PGA. After that, 3-PGA is getting converted into 2-PGA. Here the enzyme which is working over here, that is mutage enzyme. Then 2-PGA will get converted into 2-PEP, 2-phosphoenol pyruvate. Here the enzyme which is working, that is enolase, and in chemistry also you study enolation, right? Here 2 water molecules will be removed, right? After 2-PEP is getting converted into 2 molecules of pyruvic acid. So finally 2-pyruvic acid is formed in the glycolysis process. One enzyme you have to remember here, the enzyme which is present over here, that is pyruvic acid kinase. Pyruvic acid kinase enzyme is working over here, and here Mg plus 2 is working as a cofactor and K plus ion. Okay, and here also the 2 inorganic phosphate will take the 2 ADP will take the 2 inorganic phosphate, and finally 2 ATP will be formed. This is also called as SLP, substrate level phosphorylation, right? So finally we have got 2 molecules of the pyruvic acid, and the formula is C3H4O3, and it is 3 carbon compound. So 2 molecules we are taking from here to here, yes, no? So this is the structure also. Now you have understood in the complete glycolysis process that there is no use of oxygen at any place. Oxidation has taken place, but there is no use of oxygen, right? And the CO2 is also not released in the glycolysis process. It means there is no decarboxylation process into the glycolysis. Okay, so this is all about the glycolysis process. So glycolysis, this is the first step which has taken place into the cytoplasm. The second process that will happen, that is link reaction. Third process, that is Krebs cycle, and the fourth process that will be what? ATS, electron transport system. Now we do the calculation of the energy. So in the whole glycolysis process, total ATP that is formed, that is 4 ATP. Okay, and there is a loss of the 2 ATPs in the beginning of the step, that is called as investment phase, and so net gain will be what? 2 ATP. This is the net gain in the glycolysis process. In the glycolysis process, 2 molecules of NADH plus H plus is also formed. And when it go into the electron transport system, and in the ETS system, the 2 molecules of the NADH plus H plus can give us 4 ATP and the 6 ATP. It will give us 4 ATP when the glycerate phosphate shuttle system is going on. It will give us 4 ATP. When the malate aspartate shuttle system goes on, it will give what? 6 ATP. 6 ATP, right? So net gain, if you calculate, so 2 ATP this side, 4 ATP this side. If glycerol phosphate shuttle system is going on, then 6 ATP will be formed, and if malate aspartate shuttle system is going on, then total ATP will be what? It will be the 8 ATP. Okay, so this is all about the glycolysis process. Thank you.